So this talk is an introduction of general concepts that can be applied to Illumina sequencing data, but it can also be applied to sequencing data and data analysis in general. So the goal here really is to introduce you and acquaint you with concepts. We're not going to dive into any particular application or concept in great detail, but we are going to familiarize you with all of these ideas. Okay, so let's jump in. Okay, so the first portion, we are going to talk about experimental design. Experimental design has a lot of impact on our actual analysis later on, so we want to make sure that we give it the correct time and attention to make your data analysis as successful as possible. Next, we're going to dive into key bioinformatics concepts. So we are going to talk about different methods for data analysis and how they apply to specific workflows and how they can be used for your particular experiments. And then the last portion, we're going to be talking about Illumina data analysis solutions. So we'll make sure to cover all of our solution suites, both on instrument, on cloud, etc. All right, so let's first talk about our considerations for designing your Illumina sequencing experiments. So while you're designing, before you even run any of your samples on a sequencer, you want to make sure that you fully understand what your hypothesis is or what is it that you're testing. This is particularly important because it's going to drive all of the other considerations that will make our experiment successful later on in data analysis. Another very important consideration is what sequencing platform are you going to use or do you have available? As you may know, a NovoSeq will produce a ton of data compared to an iSeq, but a MySeq, for example, can run longer read lengths than, say, a NextSeq. So again, these platform ideas and considerations also become very important in how you're going to perform your analysis. And next, and also very important, is what type of analysis, including software, are you performing? Do you know of a particular aligner or variant caller that you want to use? If so, you might want to pre-check and see what software pipelines are available that include that particular tool in the analysis. So once you've completed your design, this is going to drive your planning. So questions like how much data is needed is going to be very important. How much data kind of spans, it's a very big question. Are you looking for, low frequency variants, in which case you might need a lot more data than, say, your typical whole genome type of experiment. Another important question are, will single reads be sufficient or do you need paired end reads? This again becomes important because if you're using particular software or if you're looking at a particular algorithm that you want to use, some may not do well with, say, paired end reads and may require a single, while others look for paired end reads to complete a particular analysis. And then what read length is appropriate for your experiment. This also becomes very important depending on what you're trying to do or accomplish. So here I've linked you out to an upcoming webinar called Sequencing, How to Plan Your First Sequencing Project. And in that webinar, there will be a lot of in-depth discussion on this whole concept and idea of planning and how to get ready for your sequencing project. Okay, so let's talk about how much data is required. So this is going to be one of the first pieces that you're going to need for your analysis. So here we want to consider our application, our power of detection, and our genome size. So generally speaking, if you have a smaller genome and you want to do an application like RNA expression, you're going to need fewer total reads or less data than, say, if you're going to do a whole genome sequencing experiment on a bigger genome, say like a human genome. On that, switching applications to de novo assembly, if you plan on basically reconstructing a very large genome, then of course you're going to need a lot more reads than you would a whole genome sequencing experiment or an RNA expression experiment. So let's think about this data requirement a little bit further with some examples. Again, these are just very basic examples. It's just to really illustrate the point, but I think it'll help put things into perspective. So let's consider the first two rows. We're talking about resequencing. Resequencing is your alignment and variant calling application. And we have two different species. We have human and we have Arabidopsis. Now here, we need to consider the genome size. A human is 3.4 gigabases, whereas Arabidopsis is 0.125 gigabases. So that's really kind of the big difference between these two. And then the coverage, though, we're gonna cover them exactly the same, 30x, 30 times. Generally, for resequencing, you can go between 10x to 30x, but let's just do 30x for this example. So for a human, you're going to need 100 gigabases to fully cover and complete this experiment type, while for Arabidopsis, you're going to need 3.75 gigabases. So now, understanding how much data you're going to need to fulfill this type of experiment then goes back to, okay, do you have the right sequencer? Are you using the right length, etc. Let's switch gears, but let's keep the same species, human and Arabidopsis. But this time, let's think about the application as de novo assembly. So we're keeping the same genome size, of course, and now our coverage is going to be higher than in resequencing because since we're trying to assemble these types of genomes, we're going to go for 70x. And here you can see we need even more data than we would need for a resequencing experiment, and of course, also a lot more data between the two different species. And the last example at the bottom, if we're talking about an RNA-Seq experiment, so we're checking for gene expression, in this case, we generally need less data, and we talk about needing millions of reads because we're going to eventually want to count them. So the genome size doesn't really become applicable here. The coverage will depend on the sensitivity, and again, as we're talking about, really what we're shooting for are the millions of reads number, which of course then makes this drastically different from the above two examples. Okay, so now that we have an idea about amount of data that we're going to need, let's talk about what that data is composed of, or basically reads and what they are. So a read is the data that comes from a single cluster on the flow cell. This is the actual genomic sequence found for your sample, and it can look something like this. In this example, I'm showing a 50 base pair read. This means that it would take 50 cycles from the sequencer to produce this piece of information. In this illustration here, we're just showing that as a cluster lights up, the sequencer is able to determine what base is present at a given cycle. Once it determines all the bases present and we go through FASTQ generation, we actually get reads in our FASTQ file that are coming off of those clusters. Reads are stored in FASTQ files, and these are generated after demultiplexing. So basically, our FASTQ files are going to be our input for any and almost every analysis that we're going to do from here. Okay, so now that we understand what a read is, let's talk about single reads versus paired end reads. In this example, I just chose 100 base pairs as an arbitrary length, so this is a nice, easy example. And the gray line here, it illustrates your library insert or your actual genomic DNA fragment. So in a single read, you're going to read the fragment from the 5' to 3' direction for 100 base pairs in this example. Of course, you can have various different lengths. Now, if your library insert is longer than, say, 100 base pairs, let's say it's 125, you're only going to read up to 100 or the first 100 base pairs of it. If it's shorter, let's say 90, you're going to read the full 90, and then you're going to sequence slightly into the adapter. I don't want to talk too much about this, but you do have to flank your library inserts with adapter sequence in order to sequence on the instrument. So those adapters that you ligate, a piece of it is what you would be reading if your insert was smaller than 100. So now with paired end, you're going to read that same exact library insert. You're going to read it in the forward direction for 100 base pairs. And then after turnaround chemistry, you're going to read it for 100 base pairs in the opposite direction. So now if your library insert is 200 base pairs, you're basically going to read the first 100 in the forward orientation and the ending 100 bases in the reverse orientation. So you would essentially cover the library entirely, the insert entirely. Now you could have this third situation where you have paired end reads with overlap. So now we're looking at our insert size, and now it has to be greater than 100. Let's call it 300. And if we sequence 200 base pairs in the forward orientation and 200 in the reverse, we're going to have that 100 base pair overlap. Now, sometimes we want to do this if we want to just cover the area more. That's perfectly okay. But again, this type of scenario could be problematic to certain software downstream. So just want to make you aware of these different situations that you can have with single reads and paired end reads. Okay, so now that we understand these read types, when are they appropriate? So if you're going to do SNP detection, or basically variant calling, variant identification, you can use really either single reads or paired end reads. Here, what's really important is your coverage. Are you reading the bases enough? Now, if we're going to talk about indels or identifying indels or structural variant detection, meaning when we have rearrangements when compared to our reference, then really we want to have paired end reads. We want to make sure that we have the start and stop locations. That's typically what you're looking for in your paired end reads, so that you can detect these bigger structural variations in your sample. Switching application to de novo assembly or even transcriptome assembly, again, you're going to want to use paired end reads because that start and stop information will really be helpful in allowing us to reassemble our samples. For RNA-Seq type of applications, particularly for expression, you can really do either single read or paired end. Just note here, paired end reads, again, because of if you're going to be looking for novel transcripts or if you're going to be assembling, you would want to go with paired end in that situation. And if you're looking for fusions, RNA fusions, again, paired end would be the way to go. Now, lastly here, our last application type, I have small RNA for differential expression. Small RNA, much smaller, so you're going to want to do single reads because paired end will result in high overlap, which can then break the algorithms associated with small RNA. Okay. So this last idea here in our planning and designing phase is basically selecting an appropriate read length. Of course, the most important thing with your read length is the size of your insert and what you're targeting. But just in general, these are some of the guidelines that we have. So whenever you're doing small RNA, we're looking at a smaller read length, around 36 base pairs or so. You could even go up to 50 if that's acceptable. Once you start going into your RNA-Seq, like other RNA type experiments, usually your range is going to be between 50 and 100 base pairs. You really don't expect to go much higher than that. Once you hit the 100 base pair mark, we can also apply this length to resequencing experiments. This is a good length to start these types of experiments. And then increasing our read length to around 150, this is going to be useful, again, in your resequencing experiments, but now if you're trying to find insertion deletions or any of these structural variants, switching applications to de novo, 150 and even greater can be very useful to be able to reconstruct a genome. And gene structure characterization, of course, that you want to have these higher lengths, again, so you can capture more of your sample so you can really see those differences. Okay, so now that we've basically covered the different types of ways that we can get our data, now let's talk about what we can do with our data once we have it. So I just want to go through a quick overview of what we're going to cover in this section. So first, we're going to do a FASTQ file overview, kind of an idea of what is in those files. We're not really going to talk about demultiplexing and how to generate those files, but I want to make sure that you understand what's in them and how that's used downstream. Then we'll spend some time with resequencing applications. You've heard me already talk a lot about it. This is our alignment and variant calling, but we are going to go a little bit more in depth with it, and we're going to talk about whole genome sequencing, of course, versus targeted. And targeted resequencing refers to your amplicon and enrichment type. We'll talk about that a little bit more. Then we'll do an overview of assembly applications, how they work, how they're useful, and of course, we'll talk about RNA sequencing. Now, again, this is just meant to be an introduction and an overview. There are many other concepts and ways that you can analyze your genomic data, but these are basically our most popular ways and our most popular applications that we use with our data. Okay. All right, so FASTQ files. You hear a lot about them. They're typically the input for any and all analysis downstream. So here I have on the top, I have a snippet of what a FASTQ file looks like. Here I'm actually just showing 3 reads. The first line of each read is called the header. That's marked here. And this header line gives us information about the sequencer used, the run, the flow cell, XY coordinates, etc. And then the very important piece of information here is the index. So the index is used to identify this particular read to a sample. So you can have single index as what's shown here. This is a single index 6 base pair read. Or you can have dual index. And when you have dual index, you'll see your read one, I'm sorry, your index 1 and index 2 separated by a plus sign in the header line. The next line contains your actual read or the genomic information that was read from the sequencer, and basically what we're going to use as input to everything else. There is a plus in between the read sequence and then this last line here, which is your Q score, your quality score. This line is ASCII encoded, so these characters correspond to an ASCII number that will then give you your Q score. Typically, we look for Q scores for the base calls higher than 30, but there are occasions when we have lower, and that might be still okay. Now, when you take all of your numbers of reads and you multiply them by the read length, you get the number of base pairs. So earlier when we were talking about number of base pairs needed, gigabase pairs needed, that's how you would calculate this. Another quick thing about FASTQ files, typically you don't want to open them because they're very, very big. This example here just shows us 3 reads, but they typically contain millions of reads. So we don't really recommend opening them unless you're in a command line type of environment. Okay. So now that we understand what's in our FASTQ file, let's talk about resequencing. Resequencing refers to alignment or mapping the reads to a reference genome. After we align, we then want to perform variant calling or detecting where the samples are different from the reference. When we say resequencing, we're typically talking about whole genome or whole exome sequencing. Targeted resequencing, like in amplicon or enrichment workflows, that refers more to performing alignment and variant calling on a specified region or regions of the genome. And of course, we'll make distinctions as we go along as to how we're able to target particular regions versus aligning to the whole genome. So here I just have a very simple diagram of the resequencing workflow. There are, of course, many other steps and littler steps involved, but this is the basic idea. You take your FASTQ files, you put them through your aligner. There are many types of aligners. From the alignment, which is mapping it back, we get a BAM file. We get both BAM files and BAM.bai files. Once we have our aligned reads, we then go through variant calling, various different variant callers that we can use. And from there, we get VCF files and GVCF files or genome VCF files. And we'll talk about those a little bit further. Okay, so let's talk about alignment. Alignment, again, refers to determining the location for each region where it most likely fits in comparison to the reference genome. Our output files are going to be BAMs. So we're going to get a file that will be the sample name.bam and the sample name.bam.bai. This second file, that's going to be the index file, and it helps us to, when we take these files and put them into a visualizing tool, it'll help us be able to see those locations where the reads are aligning. Now, there are several considerations when we talk about alignment. So, of course, the first one, there's millions of reads to align. So how quickly can we do this? Or do we want to take a little bit longer, but maybe have more precision? Again, these are important. Again, another consideration, you can have a read that maps to more than one position. Different aligners do different things with this idea. So they have a read that aligns to more than one position. It might either throw it out completely, or it might assign it an alignment score. So then we can decide, how we want to proceed with that particular read. Of course, with alignment, DNA features impact the alignment, meaning if your sample has variation, which is what we're looking for, we want to see how different it is from a reference, that makes it more difficult to align back. So we want to make sure, our aligners are able to handle the variation and still align it to the correct location. Okay, so here we have an example, and again, this is a very simple example of alignment. I have our reference genome here in gray, and its aligners might place a short sequence in many places in the genome sequence. Okay, now let's say we have short reads of short read lengths. We have these sequences in and en. Now, these are shorter sequences, and these can be aligned in these locations, but it can also align in these locations. So how can we, how can we make the accuracy of alignment a little bit better? So we could sequence with a longer read length. So by increasing the read length, we now have in underscore the and enome, and those can only align to one location, and that's right here. So that's good. So in that particular example, we're able to increase the read length and then increase the likelihood that we're going to place it in the correct location. Now, increasing the read length isn't always ideal. So here we have completely different reads. We have the read place and the read sequence, but now place can align into these two locations. So we have to make a decision about what comes next after place. And sequence can also align into these two locations. So does that read sequence really belong at the end, or does it belong in the middle, and there's other things in between? And so one solution here would be to do paired end sequences, paired end reads rather. So in your paired end example, you now have place, which is paired with in, meaning your read one was covering in, and then read two is covering place. And then we can safely align that to only one location, which is right here, where the in matches here and place matches here. And lastly, the last example that we want to talk about is we have this read placed. Now, placed doesn't appear anywhere in this genome because it has this D at the end. So the aligner needs to deal with basically, is this an insertion and does that belong here? Or is this a variant and does it belong here where the reference is an S, but your sample is a D? So that gives you a good idea of what aligners have to be able to do and the decisions they need to make in your workflows. Okay, so now let's talk about targeted alignment of reads. So here we just have this image of a full genome, and let's say we're only interested in two locations or two regions of this genome. So we're interested in this leftmost region, which is about 30 base pairs, and maybe this rightmost region, which is about 50 base pairs. Now, how can we ensure that we are aligning to these locations only, or that we're only doing our variant calling in these locations? We would need to make sure that we have some type of file to depict these regions of interest, and that would be a manifest file. So I don't want to spend too much time talking about the manifest file, but I do just want to make you aware that this is what's needed and it's important in your targeted sequencing. So important things that the manifest file can contain or has to contain is our reference genome. And here we're seeing this is going to be for HG19. Another important piece is that it needs to have our chromosome start and stop locations. And in this particular manifest, this is more for amplicon analysis. So we also have our target sequences and what they need to be. They're listed here. Now, for targeted alignment, of course, we have a manifest file that we're showing here, but you could also use a bed file, which you may be familiar with as well. So what does this look like when we actually align? So here I'm showing an image, and our red reads, our red lines are our R1s, or our forward reads, and our purple reads are our read 2s, and they're in the reverse orientation, and there's complete overlap, as you can see here. And basically what we're able to see and what we're able to visualize is that all of these reads are aligning to this one location, this particular gene, which allows us to, one, confirm that we are in fact doing targeted alignment, and two, if this location would be in the manifest and that it is aligning on target. Okay, so we now have a good understanding of alignment and what aligners need to do and what needs to happen during alignment. So let's talk about variant calling. As you probably already have an idea, this is to help us identify how the sample read sequences are different from the reference sequence. So you get two different output file types. You get a VCF, which stands for variant calling file, and a GVCF, which is a genome variant calling file. Now, the difference between these two is that the VCF file only contains information about the variants that were actually found. So anytime that the variant caller determined that the location was different from the reference, it's going to go into the variant calling file. The genome VCF file, on the other hand, it contains information about every position, regardless of whether it was a variant or not. So the GVCF files come in handy, particularly when you're talking about targeted variant calling, because if you're expecting, let's say, a variant at a particular location, but you don't find it, you can then look at it in your GVCF file and see what was actually called. It may have been called a reference, but you may have had some evidence of your variant. So that's when a GVCF file comes in handy. GVCF files for whole genome sequencing, although available, they're huge, so that might take more time to read through and comb through. Okay, so some considerations for variant calling. How to call the variant, basically your depth and your coverage. Did you do a 10x coverage versus a 70x coverage? Of course, that's a big difference in coverage, and will change how if your variant is called or seen or not. Then we have somatic versus germline, which we're going to illustrate a little bit further, but somatic basically means you're going to be looking for your low frequency variants, variants that are occurring perhaps in like a tumor tissue or some type of cancer research, versus germline. This is going to be more your heterozygous, homozygous, what you would expect to see in normal hereditary patterns. And then another consideration, how you're going to annotate and how you're going to interpret these variants. So the VCF, they contain all of the information about the variants that were found, but then it's still up to us as the researchers to go ahead and comb through those VCF files and determine what these variants mean. Okay, so let's look at a variant calling example. So again, very simple, very simplistic examples here. So we're showing our reference genome is in gray and our reads are in blue. And here we're seeing one variation. We're seeing a C that's different from the A in the reference genome. Now, can we say that this is, this is an actual variant? The answer is, just based on this example, we only have, a depth of 1. So we can't confidently say that, that it is an actual SNP, because it could be a real variant, or it could be a sequencing error, or it could even be a misplaced read. So it's still a variation. It's still an interesting location to look at, but can we confidently call this a variant? The answer would be no. So now if we increase our coverage, so here we're looking at about 4x coverage, and in all of these reads, we're seeing that the C call continues to persist. So in this case, because we have more coverage, we have more confidence that this is in fact a true variation, that it would be a variant, and it would so be called. Of course, this idea is appropriate for germline variant calling. So let's think about this, but now in a somatic variant calling scenario. So here we have, again, the same coverage, and we see only one read with the C difference. So if we were thinking about this as we had before for germline calling, of course this wouldn't be called because we can conclude that maybe it's an error, sequencing error, whatever that might be. But if this was now like a cancer tissue biopsy, then we want to know about this particular position because we're expecting these to be low frequency. So if you're going to look for low frequency variants, important to consider, make sure that you're selecting a somatic variant caller, and also that you know the frequency at which it can call at. Most of our Illumina variant callers are going to be in the 3 to 5% allele frequency, which would of course call something like this in this example. But if your SNP is even lower, you might want to consider, what can be done at that point. Okay, so let's now switch gears. Let's talk about de novo assembly. So now we're talking about a completely different application from resequencing, and a little less common, of course, too. But what is the goal here? We are going to sequence a novel genome and assemble it without the aid of any reference genomes. You can have an assisted de novo, but typically when we talk about de novo assembly, we're referring to not having anything or any idea as to what the sequence would be. So now your output file types, you're going to get contigs, so they're going to be FASTA files. So you're going to get your sample name .fa as your output. So some considerations with de novo, the size of your genome. Although we're doing this for the first time, you should still have an idea of what the size of the genome is going to be, either based on other organisms that exhibit similar traits to your organism of interest, or by other means. You should have some idea of what this genome size is going to be. Another important consideration, more so for the algorithm than for us, is going to be the continuity of the contigs. So basically how well we can establish that these contigs or the sequences that we're getting, they are continuous. And lastly, it's going to be the organism itself. If you have a multi-chromosome organism, you're going to have a lot more contigs, which is going to make this task a little bit more difficult. If you're considering a bacterial species where you might have a single chromosome, this process might be a little simpler. Regardless, though, de novo assembly is iterative. You are going to have to do it several times. So let's look at an example. Typically, we're going to want to do some type of mixture of long and short reads. The long reads, they're really going to serve as the backbone of our genome, where the smaller reads, the shorter inserts rather, they're going to be the middle pieces that we missed with the longer inserts. So your FASTQ files, you're going to have reads of all sorts of varying insert sizes and lengths. And the goal is to take all of that and assemble it so that you can assemble the backbone and fill in the in-between with the smaller inserts. So from here, you can then get a consensus sequence that may look something like this as your assembled genome. Now, again, this isn't going to, typically this doesn't happen in your first run, in your first analysis. It's going to take several iterations, and it may even take working with the parameter settings of the tool that you're using, but eventually this is what you're looking to get. Okay, let's switch gears again and talk about a totally different application. Let's talk about RNA-Seq. So here, the most common RNA sequencing task is that you want to measure expression across the transcriptome, or measure how much expression there is for a particular gene or transcript. So just to get a brief overview of what we're looking to do here, we are going to take our biological samples, extract our RNA, convert it to cDNA, perform Illumina sample preparation, and then we're going to sequence. So in your gene expression study, you're looking for abundance of reads for a particular gene that indicates abundance of gene transcripts present in your sample. You want to measure abundance, and since you're measuring abundance or how many reads are lining up to a particular transcript or gene, this essentially becomes a counting application. So what does that mean? It means we can take a plant at time 0, add a chemical to it or a treatment, and measure what genes are expressed half an hour, 2 hours after the treatment. So basically the idea, or maybe the hypothesis, would be that this chemical might wear off after a certain amount of time, so you might expect to see less expression as time passes, or maybe it's the opposite. But basically that's the idea of what we're trying to do. So how do we do this? So RNA-Seq analysis begins with alignment, except here alignment is different than what we would use for our resequencing applications. So if you have single-end reads, they can align to exons, specific exons. So in this example here with these single reads, we see that 2 reads are aligning to that first exon, 2 other reads are aligning to the second. We don't get any other exon alignment. Now with single reads, you can have 1 read aligning to 2 exons. So here they're denoted by the dotted line, and that indicates that this is a splice junction. So they don't fully align correctly to their reference, but they span 2 exons. Similarly, with paired-end reads, you can have your pairs spanning multiple exons or junctions. So you can have your read 1 aligning to exon 1 and your read 2 fully aligning to exon 2. Or like with single reads, you can also have, say, your read 2 spanning 2 exons. Now, this information, of course, is important because it gives us information about exon-exon boundaries and exon associations, but getting back to the task at hand, we want to make sure that we are counting these alignments correctly so that we could then get our read counts that correspond to our RNA transcript abundance. So how do we do the actual counting? Now, there's a couple of ways that we can count expression, but I'm just going to show kind of 2 simple ideas and models here. So let's say we have this scenario where we have a treated sample that has a depth of 5 and the control sample has a depth of 2. Now, let's say for the treated sample, we got 50 million reads, and for the control, we have 10 million. We need to normalize them if we're actually going to compare them correctly, because right now it just looks like the treated sample has a lot more expression than the control sample. So in this case, let's go ahead and divide by millions of reads. So we took our depth of 5 and we're dividing it by 50 for 50 million reads, and we're getting 0.1 for our treated sample. And now our control sample looks, we take the depth of 2 and divide it by 10 for 10 million, and we get 0.2. So now with this type of normalization, this treated sample really has half the transcript expression of the control sample. So even though at first glance we saw more coverage and reads in the treated group, when we normalize in this manner, we actually see that we have half the expression for the treated as the control. So normalization, again, it's very powerful, and your RNA-Seq software needs to be able to do this automatically. So in this case, we're just looking between samples. Now let's look at a different idea of normalization. Okay, so now what we want to compare are transcripts from within the same sample. So here we have 3 different genes. Each of them are different sizes. We have a 2, 1, and a 3 kb. Let's go ahead and throw in our reads. Okay, so now we have our aligned reads, and we're getting raw read counts of 4, 5, and 9. But like the previous example, this is not enough information. We need to normalize. So let's standardize by read length. So for the first gene, we have 4 over 2, 2, 5 over 1, which is 5, and 9 over 3, which is 3. So now we have normalized our transcript length within one sample. Okay, so now I basically showed you 2 ways of normalization, but in RNA sequencing, well, we want to have this occur automatically without so much thought. So the first way that this gets done is with reads per kilobase of exon per million of mapped reads, also commonly known as RPKM. And this takes into account reads, mapped, and the length of the transcript for normalizing our single reads. Our RPKM is specific to single reads. Now, we have a similar story for FPKM, or fragments per kilobase of exon per million of mapped reads. And this normalizes in the same manner as RPKM, but it takes into account that you have paired reads or you have fragments of a particular sample. The last normalization method now is transcripts per million of map reads, or TPM. And this normalization method is also being commonly found in more and more of our RNA sequencing tools. And this is now a different method, and it can be used for single and paired end reads. And really what the difference is the order of operations in which the calculation is done. So our RNA-Seq fragments can be counted, and they can be used to determine relative abundance when comparing them to these, comparing them with these different methods. Okay, so we've definitely given you a lot of information, a lot of things to consider. So now let's talk about Illumina data analysis solutions to help you perform these types of analysis with your data. So our first solution is Local Run Manager, and this is what the dashboard looks like. Local Run Manager is appropriate for our benchtop sequences. So this is going to be the iSeq, the MiniSeq, MySeq, and the NextSeq. And this has a modularized approach. So meaning that whenever there's a new module or a new application that you can try, you can just install the module and you don't have to install the entire framework. So with Local Run Manager, you can have it installed both on instrument and off instrument. Of course, if you're going to do it off instrument, your PC needs to meet all of the requirements. So here in the second image, what I'm showing are the different module settings. So we see different modules listed here, and there's specific settings that we can work with or manifest files that we can add, et cetera. And here at the bottom, I've linked you out to a couple of resources so that you can either download Local Run Manager for your off-instrument use. I've also linked you out to where you can find the documentation for it and even to the Local Run Manager webinar. So again, this tool is really nice because you can do it right on your instrument and it's free. So it's free to download, it's free to have it on the instrument. It's a great local analysis solution. So now we can switch gears and talk about our cloud solution. So this would be BaseSpace Sequencing Hub. It's commonly known as BaseSpace. And what you would need to do is with BaseSpace, you would have to log in and create your basic account so that you can gain access. And this first image here is showing us the dashboard. And you can go and click over to Apps, which is highlighted here in red. And we can see a variety of different apps that we have and that we can use. Now, what's nice about BaseSpace is that you can log in from anywhere, any computer, as long as it has internet access, and you can do your analysis. You can also send your runs from your instrument to BaseSpace and then perform the analysis anywhere else, which is pretty great. So a lot of the apps that we have here, they are going to match up with the modules that are available in Local Run Manager. And BaseSpace is also going to have third-party apps, meaning applications that were developed outside of Illumina but are hosted on BaseSpace. So again, I have a couple of resources here linked out for you. Again, just a link to BaseSpace. You can, create your account or just, browse around and take a look. And of course, we have some webinars too to guide you through going through BaseSpace and how to use it. The last pipeline that I want to talk about is Dragon. Dragon is our newest solution, and you can have it both as an on-site server, and you can also use it as applications on BaseSpace. So now, of course, if you have the on-site server, you are able to do a lot more with it than you would on BaseSpace, but the BaseSpace applications are still really nice, and they complete really fast for you to get your analysis results. So just want to touch upon some of the pipelines that Dragon handles. So there is ECL convert, which is basically FASTQ generation. Again, we didn't really talk about that, but that is something Dragon can do. On top of that, Dragon has thematic germline pipelines. These are the variant calling applications that we talked about, and then does mapping and alignment along with RNA-Seq alignment and quantification. And there's also methylation on it. Again, another application we didn't really touch on. Okay, so that about brings us to our close. So just again, we want to reiterate everything we talked about. I know it was a lot. So we had our whole section about designing and planning considerations, thinking about the amount of data that we need, the types of reads and the read lengths. Then we had an overview, of course, not completely covering everything, but to give you a nice introduction of resequencing, Novo, and RNA-Seq. And lastly, we talked about our Illumina solutions for data analysis, such as Local Run Manager, BaseSpace, and Dragon. So last couple of items I want to leave you with, we do have some resources. These first set of resources are going to be geared towards your design, your planning and designing. So your coverage calculator, method and kit selector, and tech notes are available here. I do have resources on FASTQ files and demultiplexing. Of course, we won't be able to do any type of analysis if we don't have the correct FASTQ files or correctly demultiplex our samples. So I definitely have resources here. And then lastly, I have additional recorded webinars that you can check out as well. We have BCL to FASTQ. Again, this is going to be geared towards FASTQ generation. We do have an enrichment data analysis webinar. Again, it's an introduction, but it does provide a lot of information. If you're interested in RNA sequencing, we do have an RNA-Seq introduction to analysis webinar available as well. And of course, if you want to learn more about Dragon, we also have a full webinar completely dedicated to Dragon. And if you want to just check out any other upcoming webinars, you can always go to our webinar page on our support site and check out what's coming up and register. Okay, so thank you everyone for your attention. I'll now open it up to some questions.